In class, we've already talked about the fact that predators can invoke selection on prey items. Specifically, we talked about how lions might induce greater speeds or invoke selection for greater speeds on gazelles. So that seems pretty intuitive. But consider also that plants need to respond to what we call plant predators or herbivores. And in this case, the plants can't run away, so they have to have alternative defenses. This is not the only set of challenges faced by plants. Like all organisms, plants face many challenges for survival and reproduction. For survival, plants need water and nutrients. They need to gather the materials for continued life. Further, they need to have access to enough sunlight for photosynthesis so that they can reproduce in future. Finally, they need to live in an appropriate temperature regime and also have not just make pollen and flower, but make enough pollen to either have it transported by wind or make nectar to pay somebody else to move their pollen for them. And there's a variety of other techniques plants take advantage of. Then, when they make their seeds, they have to ensure that somehow their offspring, their seeds, can get to somewhere new that's suitable for plant growth. The last piece of this puzzle, though, is that they need to survive long enough to do all of these things, and that requires that they avoid being eaten. So let's consider how plants might respond morphologically, that is to say physically, to some of these responses. Leaves that grow in shade will have more surface area typically than leaves that grow in heavy sunlight. This maximizes those shady leaves' ability to contribute photosynthesis to the overall growth of the plant. Similarly, plants that grow in poor nutrient soil need to invest more in roots and make a more complex root system to maximize their ability to get nutrients. Doing that requires that they don't have as much energy available to make shoot mass. So plants that grow in poor soil will make a trade-off and make more roots at the cost of making more shoots. Similarly, plants that face herbivory may also have to invest in resources that enable them to avoid being eaten. Here, we see that the Opuntia cactus that grow on islands that have cattle are more likely to have spines and have a larger percentage of the plants have spines than in those Opuntia cacti that grow on islands that do not have cattle. Some of this phenotypic variation is due to changes in how plants grow in different environments, and some of it is due to genetic variation. And of course, some of it is due to complex interactions between genes and environment. But specifically, we can see here in this arrowhead that the leaves that are growing that are submerged in water have a different morphology than those leaves that are exposed or emergent. The same thing is true in water crowfoot. How can we tell the difference between whether observed variations in phenotype are genetic or environmental, or of course some combination therein? What you need to do is take organisms from populations that have different phenotypes and grow them in uniform conditions to see to what degree their phenotype reflects the environment they came from, and to what degree their phenotype reflects the environment they're living in. So we'll take seeds from plants from different areas, plant them all in one uniform place, and look at the resulting plants. In this case, we're looking at plants of Vicia faba that were grown in different conditions. And in this case, we're looking at the number of damaged leaves that the plants experienced. And interestingly, what you find is that when the leaves are damaged, all of a sudden, these plants make more extrafloral nectaries. An extrafloral nectary is a part of the plant that's not part of the flower, so extrafloral, that makes nectar. And they would do that to attract ants. Why ants? Because ants will viciously defend sources of food. And in this case, if ants are getting nectar, they will viciously defend that plant. And if you've ever encountered fire ants, you know that they will viciously defend their resources. This raises the idea that there are constitutive defenses as well as induced responses. In a constitutive defense, the defense is always present. In an induced defense or an induced response, they only make it if they're experiencing some damage already. So when should plants have constitutive defenses and invest in defense even before they've worried about herbivory, and when should they only make defenses if they're experiencing some herbivory? And that leads to this simple model here. If the probability that they're going to be attacked is very high, then they shouldn't wait to get damaged to invest in defenses, because they are likely to get damaged and they should reduce how much damage they get. If the likelihood that they get damaged is very low, they may be able to save some energy by only making defenses if they start to experience damage. So that would be a circumstance that favors induced defenses. Let's consider this in a plant called plectritus. Plectritus grows in the Gulf Islands in the Salish Sea, and there's two sorts of islands. There are some islands that have deer present and some islands that have deer absent. And what we see is that plectritus looks different on islands that have deer than those that don't. Specifically, where there are deer absent, the plants grow tall and robust, and you can see these big showy flowers. Okay, where the deer are present, they grow much shorter. So these are phenotypic differences that were measured and observed between different plectritus plants on islands with and without deer, and then this was of course supported by a growth experiment in the lab that islands where deer are present grow shorter plectritus plants. So the next thing we need to know is how deer herbivory influences the plectritus growth. So we need to simulate deer herbivory. And rather than taking deer into the lab and letting them munch on the plants, we'll come up with an alternative way of asking this question. Specifically, they're going to use mechanical damage to look at simulated feeding by herbivores. It's not a perfect simulation. It lacks some of the physical details of how deer munch on plants, and it lacks deer saliva and other things that may be important in induced reactions, but it's a good proxy to ask this question. Here, in this experiment, we've got two independent variables. The first one is which island the deer came from, deer present or deer absent. Okay, so that's one type of independent variable with two levels. The second is whether or not the plants experience simulated herbivory. So, in half of the treatments, the plants are clipped at 2.5 centimeters above the soil, and in the other half, they are not clipped at all. So, there's two independent variables, each with two levels. That's a 2x2 experiment. So there are 4 treatment groups. Deer absent and clipped, deer present and clipped, deer present and not clipped, and deer absent and not clipped. Now we have to figure out what we're going to measure, and the first thing we want to think about is how that impacts overall growth. So we're going to look at what proportion of plant growth or plant height is lost due to the clipping. The second thing we're going to look at is the proportion of seeds that the plants make on lateral branches. These are branches that may not be lost when the top of the plant is lost. And we need to think about what predictions we have. So if we think that plectritus might have an induced reaction to deer herbivory, then we might expect to see some differences with the clipping treatment. If it's only strictly a genetic issue and it's constitutive, then we would expect to see only differences between the two populations. In fact, if we look at the differences in height or the proportion of height lost, then indeed the deer present populations lose substantially less height to clipping than the deer absent populations, and that was pretty easily predictable. Interestingly though, we also see an inducible reaction in the deer absent populations. So when they're clipped, they put a higher proportion of seeds on their lateral branches than when they're not. We don't see the reaction, that same inducible reaction in the deer present population. So these data suggest that there are constitutive evolutionary responses that you see because plectritus from populations with deer herbivores are better defended from herbivory by growing shorter. But further, the plectritus population that grew from a population that didn't have deer present showed signs of an inducible defense by increasing the number of seeds it produces on lateral shoots. So the next question is, how does this relate to real deer herbivory in the real world? And we can ask that question. In this case, we're going to take plants and transplant them to deer-present islands, both inside and outside enclosures that exclude deer. So we've got, again, two independent variables, deer or population of origin, deer present or absent, and whether or not they're inside or outside the deer exclusion fences. And on this, we're still waiting for the final results. But this is a good place to point out that we can't always use a manipulative experiment, and it's not always the best place to start. Many of our ideas come from observations like where we started here. Gosh, it seems like plants that grow on deer-present islands are shorter than those on deer-absent islands. And the first place to start is by literally measuring plants in those two places and looking to see if there's a statistically significant difference. So that's a good correlational start to this pathway that we've chosen to follow looking at plectritus. We can also consider this when we look at the oceanic acidification experiment proposed by Kim Geyer last week. And in this one, she talked about the effect of oceanic acidification on the growth of seagrasses. Certainly, we can take seagrasses into the lab and manipulate acidification of the tanks and look at plant growth. We can also go out to the field and look for correlations between more acidified and less acidified sites to look for differences in seagrass growth. What we can't do, or shouldn't do, is dump acid into wild places to see how that changes plant growth, right? We don't want to cause more damage just to learn about damage. So this is a case where to get a better understanding of what's happening in the field, we really need to do correlational studies. Similarly, if you're interested in the military sonar's impact on whales, we think it's damaging. We're not going to blast sonar at whales, wait to see what happens. Do they get damaged? We will instead continue to look for correlations between where the military uses sonar and what happens to the whales who are nearby. This is also the case if we consider, lead paint on human brain development and other circumstances where really our first effort is to consider the ethics of what we're asking and how we can ask questions meaningfully and move from there.